Good evening. Welcome to tonight's presentation sponsored by PWSA USA and FPWR. Tonight's topic is results of the Phase 3 beloranib BEST PWS study. Before I turn the presentation over to our presenters, let's review some housekeeping notes. Please take a moment to write down the dial-in number and access code for tonight's presentation should you become disconnected. The phone number is 631-992-3221, and the access code is 363-507-414. For those of you using the voice IP option, you may choose the option of dialing in at any time during the presentation. There is a question segment planned towards the end of the presentation. Please note that your lines are muted to avoid distraction. There's a dialog box located to the right of your screen that has an option for you to ask questions throughout the presentation, so nothing is forgotten. Your line is muted, as I said. If you do have any difficulties during the presentation or need assistance, use this question box and I will be happy to provide assistance as best I can. Please do not use the chat box, use the question box. If this dialog box is distracting to you, you can click on the arrow at the top to minimize it, then click the same arrow to open it back up to enter questions or comments. I will now turn this evening's presentation over to Jana-Lee Heineman.
Hi, this is Jana-Lee Heineman. I'm the Coordinator of Research and International Affairs for Prader-Willi Syndrome Association USA, and with me is Teresa Strong from Foundation for Prader-Willi Research, who will explain the purpose of this call.
Great, thank you, Jana-Lee. This is Teresa Strong, and on behalf of the Prader-Willi Syndrome Association and FPWR, I want to welcome all of our attendees this evening, and thank you for joining us. I think both PWSA and FPWR felt like it was important for our community to hear about the results of this Phase 3 clinical trial of beloranib in individuals with PWS, and to have the opportunity to have some of their questions answered. So our goal is really to provide a neutral forum for people to learn about the outcomes of the trial, including both the beneficial effects and the complications that arose during the placebo-controlled portion of the trial, which is what we're going to hear about tonight. So it's important to keep in mind that the FDA is mandated by Congress to consider the patient perspective in the drug development process and also in its final decision-making process when a drug is considered for approval. So we believe that, our community needs to bring a very strong and very informed voice to that discussion. And we look at this as webinars one way to provide information about this study that will help inform your opinions, we hope, and along with other information will allow you to be contributing to that discussion as we move forward. So we appreciate you joining us and learning about this. And we appreciate the representatives from Zafgen presenting directly to our community. So I will turn it back over to Jana-Lee, and I think who will introduce our presenters.
Our two presenters are Dr. Tom Hughes and Dr. Dennis Kim. Dr. Hughes has over 25 years of experience in pharmaceutical research and development. Dr. Hughes joined Zafgen in 2008 as president, chief executive officer, and a member of the company's board of directors. From 1987 to 2008, Dr. Hughes held several positions at Novartis, including vice president and global head of the cardiovascular and metabolic diseases therapeutic area at the Novartis Institute for Biomedical Research in Cambridge. In these roles, he oversaw many drug discovery and development projects targeting obesity, diabetes, and heart disease. As a scientist, Dr. Hughes led Novartis' efforts to discover and develop a drug used for the treatment of type 2 diabetes, currently approved in 70 countries. Dr. Hughes is author of over 40 peer-reviewed publications. Dr. Dennis Kim joined Zafgen in September 2011 as the company's chief medical officer. Dr. Kim is a board-certified endocrinologist and comes to Zafgen with more than 10 years of experience in the biotech and medical technology industries. Prior to joining Zafgen, Dr. Kim held multiple senior-level clinical and corporate affairs positions at Efficacy Therapeutics, a pharmaceutical company focused on the treatment of obesity. Prior to Orexigen, he was Chief Medical Officer and Vice President of Medical Affairs at Enteromedics Incorporated. Previously, he spent 7 years in positions of increasing responsibility at Amylin Pharmaceuticals. While at Amylin, Dr. Kim was also the lead physician for exenatide, which is called Byetta, and an FDA-approved therapy for type 2 diabetes, where he directed phase 2 through 4 development and commercialization. Also with us with Zafgen is Alicia Secor, a Chief Medical Officer, Dr. Narissa Crayer, the Global Head of Medical Affairs. We welcome you, and we really appreciate you spending the time explaining to our members and FPWR members the latest information on this clinical trial. I'll turn it over to Dr. Hughes.
Thank you, Janelle. Good evening, everyone, and thank you for joining us. Before I get started, I'd just like to offer a very sincere thank you to those of you on this call who participated as a patient or are a caregiver of a patient who enrolled in our BEST PWS trial. Without you, it would not be possible to advance the understanding of beloranib in Prader-Willi syndrome. I'm sorry, I am missing a page here. But just basically to say that before I begin, I need to essentially to indicate that some of the comments that we'll be making during the course of tonight's call may represent forward-looking statements, and this is because we're a publicly traded company, and that these forward-looking statements may reflect assumptions or estimates on our part that may in time change. So as many of you know, last week we were pleased to announce the primary safety and efficacy results from the randomized portion of our BEST PWS study, which is a pivotal, double-blind, placebo-controlled phase 3 trial evaluating the safety and efficacy of beloranib in PWS patients. The study achieved both of its co-primary endpoints, making beloranib the first investigational treatment to demonstrate statistically significant improvements in both hyperphagia-related behaviors and body weight. We find these results to be promising, and we're more committed than ever to advancing beloranib in PWS. Before I turn the call over to Dr. Dennis Kim, our Chief Medical Officer, to review the specific results from the study, I wanted to take a moment and let you know that patient safety is our #1 priority, and we take it very seriously. As many of you know, one study participant who was taking beloranib in the randomized portion of our BEST PWS study, and one participant who is in the open-label extension portion of the trial, who was also taking beloranib, passed away from pulmonary embolism. Pulmonary embolism occurs when blood clots block blood flow to the lungs and is now known to be a common cause of death for people with PWS. Our thoughts remain with the families during this troublesome time. While we may never know if beloranib caused these deaths, we're reviewing all of our data and working diligently with experts to better understand the risk of thromboembolic events in the setting of PWS and whether additional risk may be associated with beloranib. I'll talk more specifically about what we're doing to understand the benefit and risk profile of beloranib following Dr. Kim's remarks, but want you to know that we're committed to working diligently with FDA to design a path forward. We're deeply committed to this effort. So now I'd like to turn the call over to Dr. Dennis Kim, our Chief Medical Officer, to review the trial design and its results. Dennis?
Thanks, Tom. And good evening, everyone. It's my privilege to present this data to you today. Before I begin, I would like to reiterate Tom's sentiments and also thank those of you who participated as patients in the trial. Now let's get to the results.
Dennis?
You can go to slide 6.
Excuse me, Dr. Kim.
Tom?
Dr. Kim, you're echoing. So perhaps you are speaking in a phone and also have computer speakers on? Is there something that you can maybe do to that to prevent the echo?
Okay. I will see if I can turn off my... Is that better?
It appears to be, yes, sir.
Okay, great. Apologize for that. Okay, slide 6, please. This is the trial design for the phase 3 trial we're calling the PWS study. We enrolled patients with a confirmed diagnosis of PWS, overweight or obese, manifesting moderate to severe hyperphagic behavior, as documented by a score of at least 13 on what we're calling the hyperphagia questionnaire for clinical trials, abbreviated as HQCT. And patients were also ranging in age from 12 to 65 years. The hyperphagia questionnaire is a tool designed to assess the overall severity of hyperphagia through typical related behaviors, such as persistently asking for food and bargaining to get more food. After a 2-week single-blind placebo lead-in period, which means all patients were receiving placebo in the very beginning for 2 weeks, patients were then randomized so that they received either a 1.8 milligram subcutaneous injection of beloranib, a 2.4 subcutaneous injection of beloranib, or a subcutaneous injection of placebo, which is an inactive injection without drug, twice weekly for 26 weeks. And that's what those 3 bars in the middle of the slide represent. After the 26-week treatment period, patients were given the option to continue in a portion of the study that we refer to as the open label extension study, which is not shown on the study design diagram on this slide. Our review of the best PWS results today will be isolated to the randomized blinded portion of the study, as results from the open label portion are not yet available. As shown on slide 7, the co-primary efficacy endpoints of the study, as agreed with the U.S. Food and Drug Administration, the federal agency who oversees drug development, often referred to as the FDA, were % change in total body weight and absolute change in hyperphagia-related behaviors, as measured by HQCT total score, from baseline to endpoint compared to placebo. Key secondary efficacy endpoints include change in metabolic and cardiovascular biomarkers, including total fat mass as measured by DEXA scan, LDL cholesterol, and HDL cholesterol. Today, we will be reviewing the top-line efficacy data, which includes the data from the 2 co-primary endpoints from the 6 months of randomized portions of the trial, which are change in body weight and change in hyperphagia score. The rest of the data, including the secondary endpoints I just mentioned, will be presented at upcoming scientific meetings. Before I get into the results, I'd like to share with you some information about the patients who enrolled in the clinical study. Slide 8 outlines the characteristics of the patients in the trial. 107 patients were randomized and received at least 1 dose of study medication, whether it be beloranib or placebo. There were 6 early study withdrawals due to adverse events. 74 patients completed the full 26 weeks of treatment per the protocol, and 27 patients were actively participating in the trial when the partial clinical hold was implemented in collaboration with the FDA in October 2015, and further dosing was suspended at that point. All of these 27 patients were nearing 26 weeks of full randomized treatment period, having completed at least 20 weeks of randomized treatment period prior to October 2015. In other words, most of the patients had almost finished the trial or did finish the trial. In agreement with the FDA, we analyzed the data using a special statistical method that would allow us to account for any missing data, including those 27 patients who didn't have an opportunity to complete 26 weeks of randomized treatment. So that's the result and the statistical analysis that we'll be sharing with you today. Slide 9 provides an overview of the specific demographics of the patients who enrolled into the study. Patients were of an average age of approximately 20 years, generally evenly split between male and female patients, and predominantly Caucasian. Roughly 45% of the patients were of less than 18 years of age, And consistent with that, a similar percent of patients were taking human growth hormone when they entered the study. When patients entered the study, they had a total body weight of roughly 100 kilograms, or approximately 220 pounds, translating to a BMI, or body mass index, of about 40 kilograms per meter squared, and total body fat mass of approximately 50 kilograms. Another way to say that is that about 50% of total body mass was in fat, measured in fat per the DEXA scan. The average hyperphagia total score was about 17 as measured by the HQCT, consistent with moderate to severe hyperphagia. Slide 10 shows the PWS genetic subtypes of the ITT population, that is to say the intent to treat population, which is the population that the FDA cares about the most. That encompasses all the patients who at least got one dose of study drug. And the majority of the patients fall into the chromosome 15 deletion subtype, followed by about a quarter of the patients with uniparental disomy and a small proportion of patients in the imprinting defect category. Looking at the disease history on slide 11, we see that the majority of the patients enrolled in the trial also exhibited characteristics associated with PWS, including aggression, skin and rectal picking, inappropriate food consumption, and to a lesser extent, running away and getting sick from overeating. There were also 10 patients in the trial with type 2 diabetes. Turning now to the efficacy data on slide 12 that will show you how beloranib worked in PWS patients. For the weight reduction primary endpoint, you'll notice that those patients receiving placebo in gray displayed steady weight gain of 4.2% over the course of the 6 months of randomized treatment. This gain was not unexpected, especially given that the study did not mandate diet and exercise counseling, and these patients were not in group homes, but living in a real-world setting. Patients in the beloranib treatment groups, however, did demonstrate a statistically significant reduction in weight over the course of the treatment, shown in orange and blue, with a response observed early in the treatment period that is progressive and sustained over the course of the study. For the 2.4 milligram dose, noted in blue, patients had an average baseline weight of 105.7 kilograms, or approximately 231 pounds. This group achieved an average of 9.45% placebo-adjusted weight loss. That is to say, weight loss that's different from what placebo patients manifested. This translates to about 20 pounds in weight difference, which was statistically significant with a p-value of less than 0.0001. Patients receiving twice-weekly injections of 1.8 milligrams of beloranib had an average baseline weight of 97.5 kilograms, or approximately 214 pounds. These patients, shown in orange, achieved an average of 8.2% body weight reduction in placebo-adjusted fashion, or approximately 18 pounds, also with a p-value of less than 0.0001. So a p-value, for your information, is the probability that the results were shown as a result of a random chance. So there's another way to say it is that the study shows 99.9999% probability that the efficacy shown with drug is true, is superior to what's shown with placebo. Now I will review the hyperphagia results. As shown on the graph on the right, the mean HQCT total score for the placebo-treated arms were stable over 6 months of treatment, as shown by the gray line. In contrast, both the 1.8 milligram and the 2.4 milligram beloranib treatment arms showed highly statistically significant reductions in the score. 2.4 milligrams of beloranib, as noted by the blue line, had an average baseline HQCT score of 18.3 and achieved an average placebo-adjusted reduction in HQCT score of 7.0 units, with a p-value of 0.0001. 1.8 milligrams of beloranib in orange had a mean baseline HQCT score of 17.4 and achieved an average placebo-adjusted reduction in HQCT score of 6.3 units, with a p-value of 0.0003. Unlike changes in body weight over time, maximal improvement on HQCT's total score on beloranib is achieved by week 12, and it is sustained out to study endpoint of 26 weeks. These data demonstrate that treatment with beloranib resulted in a clinically significant benefit to patients for both hyperphagia-related behavior and their obesity. Turning to slide 14, we wanted to drill down a bit more on the results of the HQCT total score reduction to provide a better sense for the color and extent of improvement observed after 26 weeks. Each of the 9 questions comprising the HQCT is listed on the left. Individual question scoring changes are shown on the right. As a reminder, each question is scored within the range of 0 to 4 with a total sum HQCT score range of 0 to 36 because there are 9 questions. Reduction in score signifies improvement in hyperphagia-related behaviors. The decrease in scores observed with both doses of beloranib treatment uniformly occur across all 9 questions, a stark contrast to the lack of effect observed in placebo-treated patients. This further underscores the robustness and pervasiveness of the benefit driven by beloranib treatment. Now switching gears to review the safety results on slide 15, which provides a summary of the treatment-emergent adverse events. That is to say, adverse events that came on after patients started taking study drug. While more treatment-emergent adverse events, or AEs, or adverse events, were reported on beloranib, about 80% of patients, compared to placebo, about 65% of patients, the number of serious adverse events, that is to say, the most significant and notable adverse events, were comparable across the 3 treatment arms. There were also 6 patients who withdrew early from the study due to an adverse event. In the beloranib treatment arm, there are 4 patients on 1.8 milligrams and 2 patients on 2.4 milligrams of beloranib that fell into this category. The vast majority of all adverse events reported in the study were mild to moderate in nature. As previously disclosed, there was 1 death in a patient randomized to 1.8 milligrams of beloranib as a result of a pulmonary emboli. As Tom mentioned, we take this death very seriously and are still trying to better understand the circumstances of this event, as well as the second death, which occurred in the open-label extension portion of the clinical trial. Slide 16 lists the most common adverse events in this trial. Common adverse events are defined here as being greater than 10% incidence rate in the combined beloranib arms. These were injection site bruising, aggression, and hyperphagia, all generally of mild intensity and transient nature. Of these, only injection site bruising was notable as being reported more frequently in the beloranib group compared to the placebo group. There were a total of 5 serious adverse events, 2 cases of aggression, 1 in the placebo group and 1 in the 2.4 milligram group, 1 ankle fracture in the placebo group, a mental status change in the 1.8 milligram beloranib group, and 1 pulmonary embolism in the 1.8 milligram beloranib group. Again, these are adverse events that we take more seriously and are more notable because of the gravity of the adverse event. Slide 17 summarizes the adverse events leading to early study withdrawals. 4 patients withdrew due to adverse events in the 1.8 milligram beloranib treatment group. These were abnormal behavior, anxiety, mental status changes, and pulmonary embolism. This is the same pulmonary embolism that we had noted before. And 2 patients in the 2.4 milligram beloranib group, which were injection site pain and psychotic disorder. Many of these adverse events, specifically psychiatric disorders, are commonly observed as background comorbidities in PWS patients, as you all probably know already. At the end of the randomized treatment period, there were no clinically significant abnormal patterns regarding laboratory values, vital signs, such as heart rate, blood pressure, temperature, respiratory rate, or electrocardiography, or ECG, often also called EKG findings, which is just a tracing of the heart rhythm and rate recorded on a piece of paper. As previously disclosed and summarized earlier, slide 18 details the thromboembolic events reported in patients treated with beloranib versus placebo. Can we move to slide 18, please? Including 1 fatal case of pulmonary embolism, which has been mentioned on a few occasions already, in a patient who was taking 1.8 milligrams of beloranib during the randomized portion of the PWS study that was reported in October of 2015. No other venous thromboembolic events were reported during the blinded randomized portion of the PWS trial. As previously reported, a second patient death associated with pulmonary embolism in a patient taking 2.4 milligrams of beloranib in an open-label extension setting, and 2 cases of deep vein thrombosis in 1 patient in 1.8 and 1 patient in 2.4 milligram beloranib occurred during the open-label extension portion of the PWS study. No other deaths have occurred in the course of the beloranib program. We're actively working to better understand the best benefit-risk profile of beloranib in PWS and are in active dialogue with our experts and the FDA to review the data and identify a path forward for beloranib. Let's conclude with slide 19. In summary, PWS is the first phase 3 pivotal trial to show significant weight loss and improvement in hyperphagia-related behaviors in PWS patients. As compared to the placebo group, which had consistent weight gain throughout the course of the trial, both beloranib-treated groups had statistically and clinically significant weight loss. The statistically significant reduction in hyperphagia-related behaviors in the beloranib-treated groups represent a clinically meaningful decrease in this life-altering symptom of PWS. Along with the overall safety data, the efficacy results continue to inform the benefit-risk profile of beloranib in PWS. The results reviewed today represent the dedication and hard work on the parts of the patients, families, and clinical trial sites. We would not be here today without their commitment and collaboration, and for that, we thank you. Let me now turn the call back over to Tom to discuss next steps for beloranib development program for PWS patients.
Thank you, Dennis. As I mentioned earlier, advancing beloranib in PWS is a top priority at Zafgen. However, patient safety is of the utmost importance to us, and we need to first better understand the benefit-risk of beloranib in PWS patients. To do this, we're actively reviewing prior preclinical and clinical data of beloranib, as well as additional data we're obtaining from this study to better understand the benefit-risk of beloranib. We also need to work closely with the FDA to identify a path forward with the drug. As you may know, after the patient who was receiving beloranib in the open-label extension portion of the study passed away, the FDA placed beloranib on clinical hold. That means that at this time, patients cannot receive beloranib. To address the clinical hold with the FDA, we've been working actively on several fronts, which we outline on slide 21. The FDA has requested the efficacy results from this trial, ZAF311 or the PWS trial, as well as the efficacy results from a study of beloranib that we're conducting in severe obesity complicated by type 2 diabetes, which is expected to read out in the next few months. The second component that the FDA raised is the drug safety profile. The new safety data reported today, based on 6 months of treatment, increases our level of understanding of the drug and further strengthens our resolve to continue development of beloranib. With respect to the imbalance of thrombotic events seen in our clinical program for beloranib, we're working diligently to better understand thrombosis and its relevance to beloranib treatment. We're in PWS and thrombosis experts to better inform our approach to this effort. We've thoroughly reviewed our existing preclinical, meaning animal studies, and clinical, meaning human data sets, and are also conducting studies to further elucidate any potential reasons why we might be seeing this imbalance. These approaches include a variety of studies, including assessment of blood parameters, genetic risk factors, and concomitant medications that are known to impact blood clotting in patients both in and outside of our studies, and more. We anticipate having a rich context to draw from regarding thrombosis biology in PWS patients and in patients with obesity in general, and regarding any changes that might occur following initiation of treatment with beloranib, which would be very important for us to tease out whether or not there's a change in these parameters as a result of initiation of treatment. These efforts have been underway since we reported our first serious thrombotic events in October. We've been working with experts in the thrombosis field and in PWS care to develop these approaches. Concurrent with this effort, we're also working to understand the incidence of thrombotic events as drivers of mortality and severe morbidity in patients affected by PWS. Finally, the FDA has requested that we propose a potential risk mitigation strategy for beloranib in PWS to address the concerns about thrombosis. A risk mitigation strategy is a proposal that we provide to the FDA outlining how we believe we could potentially use beloranib safely in PWS patients. While we've not finalized our risk mitigation strategy yet, it may include things like special tests and screenings that patients need to receive prior to being able to take beloranib, and possibly while undergoing treatment as well. We've already begun that work in consultation with our thrombosis, PWS, and regulatory experts. We're hopeful that with the proper screening and monitoring protocols in place, potential thrombotic risk can be effectively managed. The resolution of the clinical hold will be a process, and at this time it would be premature to provide specific guidance regarding when we might have a resolution. That being said, it remains the top priority of the company, and we're actively engaged on multiple fronts to gather the data and analyses that will first and foremost ensure patient safety and provide us a path forward for beloranib in the hopes of one day receiving FDA approval for this product. We'll now open the call for questions. Thank you.
The first question reads, I'm concerned for the participants who received the high dose of beloranib. When the FDA stopped the study, did those on open label get cut off, and were their dosages reduced gradually?
Dennis, would you like to answer that?
Sure, Tom. I'm assuming that the concern is behind the pulmonary embolism. Just to clarify, that one patient who had the pulmonary embolism in the blinded portion of the trial was actually on the low dose of the study drug, or beloranib, at 1.8 milligrams. When the clinical hold occurred, we took all patients off of drug, and as you recall, after instituting some level of safety monitoring program, we initiated study drug, or beloranib, and then a full clinical hold occurred soon thereafter, and all dosing stopped. We have no evidence at this point from a lot of the clinical trials that we've run and in animal models of toxicology studies that we've done that sudden halting of the drug has any adverse effect. That is to say, we don't have any evidence to suggest that we need to gradually reduce the dose before stopping the drug. So we didn't have a need to do that. The FDA didn't ask us to do to stop the drug gradually. everything that we've done so far suggests that just stopping the drug cold turkey has no untoward effect.
Thank you, Dennis. Next question. When and how will participants in the recent phase 3 study be told if they received the high or low dose or placebo? Dennis?
Sure. Usually in a clinical trial setting of this nature with an interventional investigational drug, it's not uncommon for patients to really never find out. However, we know that this is a special situation, and study participants and their family members are extremely involved and dedicated. And so we will do our best to try to get that information to you as soon as we can. As soon as we can means that we have to lock everything down with the database and to finalize the study report, which is a regulatory document that the FDA accepts from us as being the final documentation of the study results, both on the efficacy and safety side. Once that's locked down and finalized, we should be able to release the randomization code such that you can find out and your principal investigator can find out what you were on during the conduct of the study. We think that'll happen probably within a handful of months or several months from now.
Thank you. Next question. Was the placebo a normal saline solution?
Yeah, so I mean, it was balanced with regard to its salt composition such that it wasn't causing any change in the amount of salts being injected at the time of the injection. But the placebo was essentially comprised of the excipients that were used to suspend beloranib itself, and then it had also materials added to it that would essentially present with a very similar optical appearance. So in other words, the placebo looked exactly like the suspended beloranib, and there's no way to tell one from the next. And this was something that was checked out by a number of people, as well as including the FDA. So visually, they're indistinguishable.
Next question. Has autopsy information become available yet for the first death?
Yeah, so unfortunately, we have not yet received the results of the autopsy. We're hoping to obtain that as soon as possible. We are in ongoing conversations with the study center and hope to get access to that. This is something that the FDA is quite interested in seeing, and so are we.
Next question. How old were the deceased, and how obese compared to average of other PWS subjects of that age group?
So the first individual who died in the course of the study was in their mid-20s, and their BMI was 55, so they were substantially heavier than the typical patient who we're studying. The average BMI in this study, you'll recall, was about 40. The second individual was in their very late teens and was quite typical of the rest of the study population. We can't comment on where they would fit with regard to the broader population, but with regard to the study population, this particular individual was roughly average in terms of body weight, degree of obesity.
Thank you. Next question. Are there any plans to conduct trials of subjects that are not severely obese?
So first of all, the trial that we just completed included patients who were enrolled with BMIs as low as 27. So this is closer to a normal body weight. In the future, we certainly would be very open to doing that. For the sake of the design of this first trial, we were required to study patients who are obese in order to be able to show an impact on body weight. So if we started the trial with a large proportion of people who were not overweight or didn't have a substantial amount of weight to lose, we might not have been able to see a signal, and the drug wouldn't have been approved on the basis of that. So in order to see a benefit, we needed to have a benefit to be seen. So our next studies may well include patients who are closer to a normal body weight.
Next question is, what is the likely time frame to complete the 4 steps you outlined with the FDA?
Yeah, so that's a really good question. It will be, first of all, we're moving as quickly as we can. The process that we're going through right now is to collect additional information from the plasma samples that we had banked from this trial. This allows us to go in and look with great detail at the underlying level of markers that would be impacting blood clotting, and we can also understand whether there are any changes in those blood markers that would be indicative of a change either with treatment with beloranib or with placebo or in general during the course of the trial, and this is an important part of it. It also allows us to look in greater depth at the individuals who were affected by the thrombotic events to understand whether there's anything that was unique about those individuals relative to others. And so this is a process that will take a little bit of time. We do expect this to take weeks rather than months or years. Once we have all of this information, we will be in discussions with FDA to go through the results and to talk about next steps. But it's about a 3-step process, roughly. So first, we need to assemble this information, have a conversation with FDA about it. That will lead then to potentially additional work that we need to do or additional analyses that need to be done, which would then lead us to being able to propose to release the clinical hold. So those conversations will take weeks to months, and we should expect that.
Next question. Do you have any idea of how long the risk of DVT will be a concern?
Yeah, so I think first of all, one of the things that we've learned in part through the study that was reported recently by Dr. Locher, but others, that thrombotic events are of concern in the population. We know now that in a study of 310 deaths that were compiled by Dr. Locher, 7% of those deaths were due to pulmonary embolism. This would argue that if pulmonary embolism is a consequence of deep vein thrombosis, that deep vein thrombosis may actually occur much more frequently in this population than 7%. And so this is something that we need to look carefully at, or I shouldn't say more than 7%, but just in general. And so we need to understand how the underlying risk of thrombotic disease carries itself out, how it plays out in the setting of Prader-Willi syndrome, in the setting of obesity, in the setting of broader populations. And once we understand that, we'll be able to speak to how it might be impacted in the setting of beloranib treatment and whether there's any lingering concern there. It'd be our expectation, given where we are today, that we'll continue to study this for some time, and that this will be a feature of our program that will hopefully also open up awareness within the community regarding the opportunities for greater vigilance regarding evaluation for thrombotic risk and for managing that disease as well. So these are all things that we see evolving. But in short, it's hard to say right now how long that risk will be considered either for beloranib or for any drug that's being developed in the setting of Prader-Willi syndrome. And that's it's just hard to say at this point.
Next question. Will you be looking at weight gain impacts on the individuals that had to stop taking beloranib?
Yeah, we're collecting as much information as we can on that, and once we have something to talk about, we'll share that. This is obviously an important question for us, as well as the rebound in behaviors that are associated with hyperphagia, and we'll be collecting as much information as we can. And to the extent that those data are useful, we'll share them. and let's say robust as well. We'll share them.
Next question. Will the efficacy data from patients with deletions versus UPD be broken out and the data presented that way going forward?
Dennis, do you want to take that one?
Sure. Yeah. One of the reasons why we collect this kind of information, including gender, age, above or less than 18 years of age, growth hormone usage, subtypes of genetic PWS genetic subtypes, is because we want to do exactly that. We want to see that the drug works across a swath of different types of patients, and also that the adverse events reported are consistent across the different types of patients. And so we will be doing exactly that, and we will likely be presenting that information at a scientific conference, but at a later date. We'll initially focus on the primary efficacy and safety results, and the sub-analysis which the question speaks to will be followed soon thereafter.
Thank you. Next question. To the extent you didn't address this in your formal remarks, can you discuss what the process in terms of time and additional trials, if any, for getting FDA approval from here?
Yeah, sure. So, you know, the drug approval process has a lot of steps associated with it, and first, with regard to things that don't relate to the clinical program, there are a number of stages of validation of our program that need to go through review. One, for example, is that we need to be able to go through demonstration that our drug manufacturing is consistent and of high quality, and it yields to a stable drug product that's very reproducible. That's one work stream that's going on and needs to be completed and evaluated. There are some additional clinical studies that need to be done unrelated to efficacy that, for example, would relate to understanding the abuse liability potential of the drug. In other words, does it have addictive qualities to it, or does it lead to dependence? Similar to that, we may need to evaluate the impact on cardiovascular function directly in patients who do not have Prader-Willi syndrome. I'll just add that that other study I mentioned probably would not involve Prader-Willi syndrome patients. Beyond that, there are ongoing toxicology studies, including assessment of potential carcinogenic risk of the drug. This is a standard type of a series of experiments we're running. So all of these things are going on, and they would come together with our clinical program to provide a full package, which would also include a discussion around conditions under which we would control use and distribution of the drug. So all of these work streams need to go on. Now, having said that, it's unknown at this moment as to whether we would be required to run additional clinical trials and what the nature of those trials might be. It is our expectation going into this best PWS trial that we would need to run a second well-controlled trial that would be in large measure run in Europe to gain experience in the language and the countries over in Europe. So that trial or a trial similar to that is part of our planning. But basically, we need to run the full breadth of activities that I mentioned earlier and complete our trial program before we'd be able to submit for approval. We'll know a lot more about what that program will look like after we've had conversations with the agency, with the FDA, later this year as we move through this process. And once we know for sure what that program will look like, it'll become communicated very clearly and openly, as this is something, even from a company perspective, is quite material and very important to be communicated clearly. So as soon as we have clarity on that, we'll share that openly.
Thank you. Next question. What impact have the participants that were in fact receiving the drug had since stopping the drug? Has there been a return of all PWS behaviors?
Dennis, I'll let you answer that.
Yeah, so as Tom mentioned before, we have a subset of patients who we're following through the safety protocol that we're still running in following patients up, mostly for safety reasons without continuing to give drugs or study drugs, specifically beloranib. So we do have information on a small subset of patients, but for the majority of patients, we do not have that information because they've not come back to us to give us that information. We do get a lot of anecdotal reports that patients are gaining weight or their behavior has gone back to how they used to be before taking beloranib. But again, this is anecdotal and not systematically collected data, so we can't put a lot of stock in it. But I think the indication we're getting is that patients are, generally speaking, returning back to their baseline behavior and body weight.
Thank you. Next question. Was there no indication prior to this phase of the study of any risk of thrombotic events or animal deaths caused by embolism?
Yeah, so first of all, with regard to the animal work, we've completed most, if not all, of the animal work that's required for drug approval. It's a lot of work, and in none of those studies did we observe any suggestion of any change in blood clotting or thrombotic events or thromboembolic events. Furthermore, I would add that there's no specific mechanistic reason why the drug should cause blood to clot, especially at the low dose that we're administering, very low dose, and the exposures that we're getting. So there's nothing coming from the animal studies that would suggest that. In our prior trials, we've seen a range of thrombotic activities, but they've been very, very infrequent. So out of 500 patients, over 500 patients evaluated, we've seen up until the time that we started the best PWS trial, we saw 4 events of very varying severity. Mainly, at least half of them were very mild to moderate intensity, and they weren't considered to be of note. And accordingly, the investigators involved in this study didn't associate them as being related to study drug. So there's no suggestion to us that there was something really to be focused on here. And although we watch it as we watch all things, we know that the thrombosis is a problem of specific interest in the setting of obesity in general. And it's not surprising to see patients who are ill and have severe obesity, and in some cases have a history of venous issues to have blood clots. And so it's not a common finding in our data package so far, and it's something that wasn't of concern before.
Next question. I noticed there were 3 SAEs of patients with implanted contraceptives. Did you see these as a restriction for use in the future?
Yeah. Dennis?
Yeah, at this point, with the number of patients on contraception and the number of SAEs that we're seeing, if the question is referring to serious adverse events across all different system organ class, so we saw a few cases of psychiatrically related serious adverse event. We saw abnormal behavior, aggression, ankle fracture, pulmonary embolism. There's no indication at this point that these are related to contraception usage, with the exception of perhaps venous thromboembolic disease, which is a known phenomenon that hormone-based, especially estrogen-based contraception is associated with venous thromboembolic events. With that exception, we don't have any evidence that that any of these adverse events or serious adverse events are driven or potentiated by the presence of those contraception usage. So I don't think so at this point, but that's all to be discussed and evaluated more carefully with the FDA.
Thank you. Next question. So the PWS patients who participated in the open label will not be able to continue the study. Is that correct? Dennis?
Yeah, so I can't say yes, that's correct, or not, no, it's not correct, because we still have to have discussions with the FDA with this data package in hand as to how, if we can reinitiate dosing in patients. And depending on how that discussion goes and what's required to get there, it's possible that patients who are continuing in the open label extension right now may be able to go back on drug. But a lot of things has to happen for that to occur. So it's really up in the air, and it's a work in progress.
Next question. Were both individuals that died of the same sex? If so, is that being looked at as a factor for embolism development?
Yeah, so they were not. In fact, there is a male and a female, and so we're not looking at gender as a potential contributor to that. On the other hand, we are looking very, I would say very broadly at every possible discriminating factor. In other words, anything that could potentially pull these individuals out as being unique to the study and to their history as well. There are two things going on here. The first is that they did experience a thromboembolic event, and the second was that they were severe enough that they led to mortality. So understanding that there may be individual contributors to each of those two aspects of the events is something that we're looking at, as well as digging extremely deeply into to all of the blood chemistry and coagulation parameters in these patients, as well as the full patient population in the trial. So we're looking at everything, any possible reason we're evaluating without question.
Next question. Would you consider running any further clinical trials in Australia?
Sure. Yeah, I mean, so actually, it's like a little fact about Zafgen. We have a subsidiary in Australia. We've run many trials in Australia. The trial we've just recently completed or stopped, I should say, we finished in obese patients with type 2 diabetes, was fully conducted in Australia. We love Australia, and we're actually quite familiar with a number of experts in the Prader-Willi syndrome space who in the community working in Australia. So it's something we certainly keep our minds open to as we move along.
Next question. Did any of the subjects undergo a regular exercise program? Is there any plans to isolate a subset of subjects that does have a regular exercise program? Dennis, I'll let you have that one.
Sure. So we didn't institute a regimented diet or exercise program in the trial. Patients may have engaged in such a diet restriction or exercise regimen at home, but we did not collect that information in a systematic way, partly because it's been shown in the past in a trial setting or a clinical study setting that collecting such information is not very helpful because there's high degree of inaccuracy in terms of recall for how much people have eaten and how much exercise they've done. So it's something that we could do in the future, but it's not something that we did for this study. But it's my guess and my conjecture as a physician and an endocrinologist that I think any diet and exercise regimen added onto the drug should potentiate the drug's effect. We don't have the data to show that, but I think it's hard to imagine that would adversely affect outcome.
Next question. does Zafgen plan on reaching out to clinical participants or caregivers to see if there is a long-term impact after drug usage, good or bad? Dennis?
Sure. I think our plan all along has been to follow patients for almost indefinitely in this original setting. Because of the clinical hold, we've ceased dosing patients with any more drug. We've asked patients to continue to come back and give us safety information and to make sure that they're doing okay. And understandably, not all patients were excited about doing that because a lot of patients had to travel from far away to come to the clinical study sites. So we do have some patients who are continuing to do that, and we can collect information about long-term implications or lack thereof of the initial drug exposure. But I think it'll be probably incomplete data set at this point because of the partial nature of the overall population that we still have access to. What if and when the clinical hold lifts and we can start running studies again, our plan is to definitely continue to follow patients long term so we can understand not only the 6-month, 1-year effect, but much more time beyond that.
Next question. Are you aware of any additional DVT or PE cases following the study hold, and how long were participants followed? Dennis?
Sure. Since we've stopped dosing with study drug, we have found a few cases of DVTs, as you saw in one of the earlier slides that we presented. It's unknown to us if that DVT was pre-existing or came on after we stopped the study drug. And we continue to follow these patients to make sure that they're doing well and they're being treated appropriately as clinically indicated. And as for the follow-up for other patients, I think I already addressed that. We do have a subset of patients that we're still following that have continued to volunteer to be followed and to come to the study site. But many of the patients have dropped out and have decided not to come back because, again, the work involved and the dedication involved in continuing to do that without any perceived benefit of getting a study drug in the future.
Thank you. Next question. I understand that the results are statistically significant. In your experience, are the results medically and economically impressive enough to warrant continued excitement in the PWS and investment world, such to drive the drug to clinical availability?
Yes, that's a really great question, and clearly something that's very central to our thinking. I'll ask Dennis also to comment on the clinical relevance, but what I would say is that the results of the study, I think, really to us were very supportive, we think, of continuing development of the drug. We understand that that weight gain in the setting of Prader-Willi syndrome is a real issue. It's one that inevitably for many patients will lead to them reaching a weight that is of dire health consequence to them. So our hope was at the very least that we could slow the rate of gain during studies of this type. And we were actually very pleased to see that beyond just suppressing weight gain, that we were able to lead to weight loss in the setting of no additional control over food intake. What we also know from the course of our time working with this particular drug is that one of the stronger determinants of rate of weight loss that we've seen in the trials has been the underlying muscle mass or lean body mass that patients have had. And such that more muscled, heavier, larger, taller patients tend to lose weight a little bit faster. As I'm sure you're all aware, people with Prader-Willi syndrome have reduced muscle mass in general relative to other folks, and as a result, they have less of a furnace, basically, if you will, to burn the fat that would be liberated by beloranib treatment and driven toward fat oxidation. So we've expected to see a more modest weight loss or rate of weight loss in this population, and that's exactly what we got. So having said that, the standard or the benchmark, if you will, for drugs that are used to treat people without Prader-Willi syndrome who are obese is to reach at 1 year of treatment to reach a 5% weight loss. So for us to see 5% weight loss, in fact, a 9% difference from placebo after 6 months of treatment was for us a very remarkable and strong finding. What we would also argue is that with regard to the hyperphagia-related behavior impact, this is uncharted territory for the most part. In all of our trials with beloranib that we've run so far in patients who have not had Prader-Willi syndrome, we've heard, in the course of our data collection, we've seen very repeated and reproducible reductions of hunger, and we believe that this is a direct consequence of the drug's action on the tissues of the body. Now, in the setting of Prader-Willi syndrome, it's very difficult to get a clear understanding of hunger. In fact, it's hard, as we understand it, to really know what hunger represents in that population of your children and your family. But the hyperphagia-related behavior questionnaire developed by the group at Vanderbilt is the best tool that we had available for it. So from our perspective, we think this is a very important finding, and it's one that has a magnitude of change that we think is quite impressive and reasonable. We did speak with a number of investigators in the Prader-Willi syndrome community, and many of them were investigators in our trial. And maybe, Dennis, you could just comment on their reaction to this, but in truth, we would prefer that a lot of the discussion of that be held for discussion within the community and not be driven by the company. But Dennis, maybe you could just comment on some of the feedback that we received.
Sure. As we've alluded to before, first of all, the results are unprecedented in that no other investigational product or drug has been able to show this level of improvement. That's one thing. The other is what Tom was getting at, which is we talked to experts in the field about our results and the extent to which they change. And it's been unanimous agreement amongst all the investigators and experts in the field that this level of change and improvement is something that they've never seen before and is quite meaningful and significant enough to translate into a lot of benefit for patients if they were able to continue taking drugs.
Thank you. That will conclude our Q&A, and I'll now turn it back to Tom for some closing remarks.
Okay, great. Thank you, Sean. So first, I'd like to thank everyone again for joining the webinar tonight, and in particular to thank the PWSA and FPWR for hosting the web conference. It means a lot to us to have the interaction and support of both of these fantastic groups, and we really do thank you. We are very excited about the data, and it reinforces our confidence in the beloranib program for PWS. And we remain committed to advancing beloranib in PWS and are working diligently to resolve the clinical hold for the drug. And we really look forward to coming back to discussing how that's going with you in the months to come, and speaking with you at the various meetings that we'll all be attending. So it will be a very interesting many months ahead of us, and just please know that we are absolutely committed to doing our very best to bring the drug forward and to understand its safety and to be able to continue to use it in a way that is both appropriate and that brings benefit. So with that, let's close, and please enjoy the rest of your evening, and I'll now turn it back to Julie from PWSA USA. Thank you.
Thank you very much. I know tonight's attendees, as well as those that are watching this in a recorded fashion in the days ahead, join me in thanking the Zafgen personnel, Dr. Hughes, Dr. Kim, and all of them for their dedication and their perseverance in this clinical trial, which has had some really wonderful results for our families. I will point out that tonight's webinar, as well as the slides that accompany it, will be available for 2 weeks. All those that attended, as well as those that did not, as long as you registered, will receive a link by email within 24 hours to this recorded webinar event. Anyone on the presentation tonight, anyone viewing it in a recorded fashion that has a question that was not addressed tonight is free to send that question to me. My email is in all of the reminder emails about this, jdority@pwsausa.org, and I will see that the Zafgen personnel receive that question and can respond to it appropriately. Again, thank you so much for your attendance. Thank you to our presenters, and I hope everyone has a nice evening. Good night.